Hi, everyone. Thank you for joining us today at the Radcliffe Institute. And let me just go right ahead and move in to what we'd like to be talking about. So we have a wonderful program today on artificial intelligence and the future of health. And to help us gauge the audience, before I introduce you to the topic and the speakers, let me introduce you to me. My name is Alyssa Goodman, and I am one of the faculty co-directors at Radcliffe for Science. And I'll be your host for today. Before I continue, I'd like to acknowledge the Melanie Mason and David Niemiec Fund for Science that's supporting this event, and to remind the audience that, as with most Radcliffe events, we're going to be using the Zoom Q&A feature where you can submit questions at any time. We'll take questions. We'll have a long discussion section at the end of the presentation. And I just ask that you keep your questions as short as you can to increase the number of questions we can try to address. And so the general plan for the event is that we will have two speakers, Dr. Casandra Mangroo and Dr. Regina Barzilay. Casandra comes to us from BenchSci in Canada, and Regina comes from all the way down the street at MIT. And I'm hoping that they will introduce themselves in terms of their background and their interest in these fields in their remarks, because we have such a short program today that I'm going to actually refer you to the web and to the listing of this event for their incredible bios. I'm sorry to not include all their wonderful prizes and accolades and accomplishments, but let me just tell you that they're great, and you'll see that from their talks. And so the idea is that Casandra will speak first, and then we'll pass it on to Regina, and we will leave plenty of time, about a third of the program, for discussion amongst the three of us, and also using questions from the audience. So like I said, please use the Zoom Q&A feature for that. And so let me say that we will have two slightly different topics in terms of AI and health. Casandra Mangroo will talk about, as you see here, accelerating preclinical R&D with AI, and Regina is going to talk about AI for drug discovery. Of course, there are many, many other ways in which AI can be used in health, some of which actually our speakers today have also worked on. So some of that might come up in the discussion, but for today, we'll try to keep it to those two topics. And so I just want to make a couple of opening remarks, a couple of substantive opening remarks to just kind of get us all on the same page. So the way I think about what's different about AI, I tend to think about this in the context of a class and a project that I've been working on at Harvard for several years now that's about the history of prediction, that ends with the kind of modern work that you see here, but actually starts with Mesopotamian sheep entrail divination. And so if you look at where we had gotten with prediction by the 20th century, we kind of had filled out this whole rainbow where people notice a phenomenon, they observe it or experiment it on purpose, they collect data about it, and then they maybe come up with some kind of empirical rule, say Mendelian genetics, for example, that describes how things work. If you're really lucky, they come up with a theory, and that theory or the rule has explanatory power and can potentially make predictions. So this, as we call it, Padua rainbow, you can ask why later, kind of describes how 20th century science worked. The possibility is that now in the 21st century, we can kind of have the first part of this rainbow: phenomena, observations, experiments, data. And if we want predictions, we actually can skip rules, theories, and explanation, which is a little bit, scary, but fascinating that you can do this. And I put AI/machine learning because, as I'll show you on the next slide, machine learning is just one form of AI and not even necessarily the only one that you'll hear about today, but it's one that you hear about all the time. So I'm going to just use it as an example to show you what I mean about how you skip from data straight to a prediction. And this is the last slide, and then I'm going to turn it over to Casandra. So what is machine learning? This is a very, very high level summary. So for the experts in the audience, please forgive me. Okay, but you've heard this word training data very often. And so let's use this example of image recognition, which again is not what we're talking about today, but it's an easy example to explain. Here's some cats and some dogs. That's obvious to all of us because we've seen many cats and many dogs. We've been trained and we don't know how we recognize that it's a cat or a dog, but we do, okay? And so this learning algorithm is kind of our brain in human learning and human intelligence. In artificial intelligence, it's some magical predictive system that's fed this training data. And you're going to hear about these systems today in the talks. And so the training data goes into this learning algorithm, and there is a predicted answer, which is either dog or cat. And some of what we're going to talk about today is that the answer is only dog or cat. Sometimes there are probabilities associated with that, but usually not. It's just a decision. And there's potential uncertainty in that decision, and it's very hard to quantify that uncertainty. And some of the uncertainty is potentially caused by insufficient amounts of data or biased training data. Notice I put 4 cats and 2 dogs. So if you have too much of one kind and not enough of another, you're better at predicting some things than others. You also might make biased decisions. And then one thing that I know that one of our speakers at least is going to talk about is the difference between supervised and unsupervised learning, where you do or don't take usually human expertise or some awareness of the mechanism being simulated or predicted into account. And this will hopefully all make sense, a little bit more sense after you hear these 2 talks, if it doesn't already make sense to you. But really the part to appreciate is that you should look out in what we hear for these kind of concepts. What's the training data? What's the learning algorithm? And how sure are you about the predicted answers? So I'm going to turn the floor over now to Casandra Mangroo, and I'm very excited to hear what she's going to say about accelerating preclinical R&D with artificial intelligence. And she'll speak for about 20 minutes. Thanks.
Hello, everyone, and thank you, Alyssa and the Radcliffe Institute for inviting me to speak today. Very honored to be speaking alongside Regina. So I'm going to share my screen very quickly. Okay, so a little about me. My name is Casandra. I'm the VP of Science here at BenchSci. I've been with the company for 4 years. Prior to that, did my PhD research in the field of virology. A little bit about BenchSci. We are a startup based out of Toronto, Canada. We're about 150 people. And we work with 15 of the top 20 pharmaceutical companies. Our investors include Google's Gradient Ventures. We're actually the first company that Gradient has invested in Canada, as well as Fidelity's F Prime out on the East Coast. So I'm very excited today to share with you the technology that BenchSci has developed and the application of artificial intelligence to preclinical R&D. The goal is really to demonstrate that with the right development of algorithms, that with the right training sets, and with addressing the right problems, AI can have an incredible impact on healthcare. So let's start with the problem. So in preclinical R&D, $150 million is invested in each asset or drug with 5 to 10 years of preclinical research and development invested. But 9 out of 10 medicines that actually make it to clinical trial fail. Well, why is that? Well, biology is very complex. A disease is often not caused by a single disease or gene. It's actually a complex interactive network of proteins and pathways. And scientists spend a lot of time trying to understand those underlying mechanisms. And preclinical experiments are key in guiding the discovery and development of these medicines. And in working with our pharmaceutical partners, we've learned that it takes over 7,000 experiments and 5 to 10 years of work to really understand what causes a disease and finding the right drug to bring to clinical trial. However, scientists face many challenges at these stages. We learned that only 20% of those experiments are actually advancing medicines. And this is due to the lack of tools and resources that scientists have to make more informed decisions about their experiments. If we could eliminate those unnecessary 80% of experiments, we could help get novel medicines to patients years faster. Fortunately, the information that scientists need to determine this is actually within various sources, the first being millions of scientific publications, but that information is often buried. The tools that scientists need to perform those experiments buried across hundreds of different vendor websites that hold those different products, and key insights buried within complex scientific images. And the tools that scientists have today to actually interrogate this information are often limited. They're keyword-based, like Google or PubMed, and the results returned are often in the millions, where scientists have to manually go through all of these different results to find the most relevant information for them to decide what is the best experiment to do. And that's where BenchSci really comes in. Now, there's a lot of great machine learning algorithms that have been developed for the purpose of identifying appropriate targets for drug discovery. And on the other side of the drug discovery process, applied to clinical data. But there are really no tools to address the challenges that scientists are facing during the experimental design process to actually biologically validate those targets and make sure that we're choosing the right drugs. And BenchSci helps scientists answer the most important questions. What are the most impactful experiments to conduct? What is the most efficient way to perform them? And how can we increase the reliability for informing clinical trials? And how we do that is we developed artificial intelligence algorithms that were trained to read scientific data like a scientist would, extract key experimental entities that are important for scientists to make decisions, and surface that on very intuitive applications that now brings that data to the fingertips of scientists so they can design their experiments, get more successful results, and bring treatments to patients faster. So a little bit under the hood, how did we do that? Well, we pulled together a diverse team of biomedical scientists, bioinformaticians, data engineers, machine learning engineers, and biomedical data analysts, leveraging the relative expertise to decode the world's experimental knowledge. We took a three-step approach to do this. The first was to collect and curate that data. Often these data sources are across many different databases and repositories that scientists have to search from, including documents like publications. The tools that scientists need, like reagents, are often buried in different websites by different vendors. And of course, there's a challenge around the ambiguity and nomenclature of the different proteins and genes that we've addressed by building out a bioinformatics ontology. Now, once we have all the data sources, we can now apply our machine learning algorithms. So we use both text-based and image-based machine learning algorithms. And then we can contextualize that information using tagger models to extract the surrounding experimental context that's important for scientists as well. So in its entirety, we can understand protein targets being studied, applications being used, link that back to the figure which represents the experimental output, understand the cell lines, model systems, tissues, diseases that are being studied, all of which are key information that scientists need. So in terms of the different data sources, we access published data from both open and closed access publishers, preprint data like from bioRxiv and academic theses, third-party data sources like Human Protein Atlas and ENCODE, which are independent institutions that are testing and validating the tools that scientists are using, as well as vendors that are testing their own products. All of these sources are data sources that scientists need in order to make important decisions. Next, we had to pull together all the different tools that scientists are using. These include antibodies, proteins, RNAi, CRISPR, which many of you may be familiar with, and model systems like cell lines. Our database currently consists of over 32 million products from over 300 different vendors. Scientists searching this information often have a very frustrating time of it because this information is not standardized. So we have a team of scientists that actually pulls in these vendor catalogs, looks at the gaps, fills in those gaps, finds inconsistencies, corrects those, and make sure that those information are also updated on a regular basis. This is no trivial task. By pulling all of this information together, scientists can now search this more effectively and find appropriate products. The third challenge that we had to overcome was actually addressing the ambiguity in protein nomenclature. So I have an example of that on the left-hand side here, PD-L1. It's a common protein studied in immune checkpoint therapy in cancer. It's also known as CD274, B7H1, amongst the others listed there in that table. So scientists can use any one of those terms when they're citing it in their publication or in their research, and a vendor, any one of those terms when they're citing it as the target of their product. So you can imagine how frustrating it is for a scientists to search each one of these individual terms, keep track of all those results. For us to apply our machine learning algorithms, we created a bioinformatics ontology that consists of over 50 million different synonyms across 170,000 different genes, across thousands of different species to address this. This allows us to ensure comprehensive data mining, and also we leverage this bioinformatics ontology to ensure the searchability of this data on the BenchSci platform. Now that we have all of our data sources, we can now apply our algorithms. So we use NLP or text-based machine learning models, as well as Vision ML or image-based machine learning models to extract data from these sources. These are all contained within a very robust machine learning pipeline consisting of over 100 different models that analyze and extract relevant experimental data entities, the protein, the reagent, the application, the figure, looking at the images, extracting surrounding experimental context from millions and millions of pages of scientific research. Now, your machine learning algorithm is only as good as its training set. And when we started out 5 years ago, we realized that off-the-shelf training sets did actually not meet our needs. So we trained our algorithms ourselves in-house. We had a team of 40 PhD scientists labeling millions of data points of text and image. Each of our scientists have domain expertise in the various therapeutic areas, as well as expertise with the individual reagents. We developed internal tools for us to be able to do this at scale, so our scientists can also evaluate the ML outcomes. And one of the other important things about this as well is ensuring that the training sets are large enough to eliminate a bias as much as possible. So the first application of our ML was text. Now, scientific text is not like regular text. There are often run-on sentences. If you look at a figure legend, there is no punctuation. Protein names are referred to in many different contexts throughout the paper. So we actually developed over 40 different biomedical entity recognition models to parse through these publication texts to detect the protein, the relevant text that refers to the experiment, the reagent, the application, and most importantly, linking that back to the image, an example of which I have on the right-hand side here, which is the experimental output. And scientists look at this information to determine the quality of the experiment that's been done. Now, we started with text, but we soon realized that text was not enough. If you read through the text alone, you'll see often references from scientists saying that 10 protein targets were studied as indicated in the image above. However, they never mentioned those protein targets or any other context, and that information is actually buried within the scientific image itself. So I have an example of one of those complex scientific images on the right-hand side here. In order for us to ensure that we collect data comprehensively, we developed semantic segmentation models that can actually help us decode those complex images, find the individual panels where those experiments are being reported, denoted by A, B, C, D, E. And within those panels, there are subpanels. So our models are actually able to detect the panel bounds and able to classify those images into the respective applications. So for example, those middle 4 images, we can determine our IHC. We can also identify graphs that are happening at the periphery. We use text detection and OCR models to extract the embedded text so we can understand the protein target, the cell lines, and other tissues that are being used, not present in the text below. We use super resolution models to enhance the quality of the image so we can apply our machine learning algorithms, and to ensure we have robust training sets, we use synthetic images to bump that up and also to ensure that there isn't any bias in our image training sets. Now, by analyzing both the text and the image, we can now understand a scientific publication as a whole. We can also apply those pipelines independently to analyze only text or only images. Next, we can apply our context tagger models. So much like the bioinformatics challenge, cell lines, cell types, tissues, diseases, species also have many synonyms and aliases. Diseases especially can have acronyms or full terms. So we've developed ontologies around that as well, where we leverage our algorithms to extract that from the text and the image. And now we can create essentially a roadmap of that experiment. Each of the individual entities from the experiments representing circles or nodes on the image on the right, and creating relational associations to understand the experiment in its entirety. To date, we have over 23 million experiments for scientists to search from. Now, before the data makes its way to the platform, the accuracy and integrity of that data is very important to our scientists developing drugs and also to us. So we want to make sure that that data meets a certain quality threshold. So we have a team of PhD scientists that are reviewing that data to ensure scientific accuracy. We also have a constant feedback loop. So with the tens of thousands of scientists that are using this across academia and industry, they're providing us feedback. We incorporate that feedback into our models. We do regular data runs, running everything from scratch and replacing it to make sure we're incorporating those improvements. And this feedback truly guides the accuracy, expansion, and capabilities and integrity of our data. Now that we have our data within our system of record, it can now be surfaced to our scientists through our AI-assisted reagent selection platform. Now, this is a platform that scientists can now use to search the protein target of interest, leverage the filters and our ontologies on the left-hand side to quickly narrow down their results to the products that are used in their specific experimental context, make informed decisions leveraging the knowledge of the scientific community to do more successful experiments. And I'd like to pause here to actually share a story, a success story of our applications, one of my favorite stories to share. Oftentimes we'll see that scientists will purchase 10 to 12 different products just to test it and find the one that can work in their experiment. And that takes a lot of time. So we spoke to one of the groups, and they shared a story with us from the past where they spent 12 weeks testing 8 antibody products, only to find one that worked in their specific cell type. So we went back to them and said, how can BenchSci help? So they input, as you can see on the interface here, their target of interest, their specific cell type. And in that situation, the result returned in 30 seconds was one product. And that one product was the same product it took them 12 weeks to find. And that's just one of many success stories of how machine learning can be applied to the preclinical stages of research and development. And our goal is to empower scientists with machine learning so that we can shorten that period and ultimately get drugs and medicines to patients much faster. So thank you, everyone. And if you want to learn more about avoidable experimental expenditures I spoke about today, you can go to the BenchSci site and look at our resources. So I'm going to stop sharing, and I'm happy to take any questions.
Yeah, that was fantastic, Casandra. So we're going to save the audience questions to the end. And I've been reading through the questions that they have, and there's a bit of a thread that also occurred to me. So what you're doing is obviously miraculous and useful. And what people are really curious about, and what I'm curious about, is you mentioned PhD scientists and experts several times. And so they're important in establishing the right training data, establishing that there's no bias, establishing that the answers come out looking sensible. But where would you say that they're the most important? And let me just say that one question from the audience really concerns tagging. And what you're doing is, I think I know, is having people tag examples, and then the learning is doing the actual tagging of the millions and millions and millions of records. And so just to clarify that as well. So I guess my question is, where are the humans the most important in that whole process, do you think? Or is it everywhere?
I would say everywhere. At the initial stages, we have scientists that are labeling that data, scientists that are checking the output, scientists that are ensuring the accuracy of those connections and relational associations that we're making. Now, a good practice is to ensure that the training sets that you develop are not the training sets that you're using to test the algorithm. So that's something that we ensure happens. We split out our training sets and make sure that we're training on a subset, and then we're testing it on another, and then we evaluate the output. So that's super important to ensure the accuracy.
Thank you. That's perfect. And just in case anybody joined late, I just want to say that we are collecting questions in the Q&A. I'm sorry that there are so many, I'm grateful that there are so many people here that we don't have the regular chat open, but please feel free to put your questions in the Q&A, and we'll try to get to as many of them as possible in the discussion section that we're going to have after we hear from Regina. So I thank you again, Casandra, and I turn the floor over to Professor Regina Barzilay of MIT.
So hi. So first of all, thank you very much for having me over. It's my great pleasure. I'm just located physically, maybe 15 minutes walk from Radcliffe now, but it seems to be like, miles away. So no matter where you are, in the world, we are all now at the same distance. So I'm going to talk today about, AI for drug discovery, really focusing on the discovery aspect itself. But before I go into this topic, I just want to say that, I'm a professor of electrical engineering and computer science at MIT, but one of my duties within the institution, I'm also AI lead for JClinic. This is a center at MIT which kind of combines effort in AI and health. And as part of this efforts, we are organizing on April 12th a conference, which is called AI for Healthcare Equity. It's free and open to the public. It's concerned to all of us to make sure that the clinical AI tools that we are developing actually work on different people. So we have a very exciting program, and I hope some of you would attend it. But now I'm going to switch gears and really focus on drug discovery. Okay. So as Casandra already kind of alluded in her part of the presentation, today the business of drug discovery is very expensive business. As time goes on, it takes more time to find useful drugs, and there are lots and lots of diseases for which we don't have, good therapeutic solutions. So the question that, people who are not in pharmaceutical industry, and actually myself, when I started working in this field a few years back, I was wondering, what's the big deal? So the big deal is that the space of possibilities is humongous. Even if you're looking at small molecules, and this is just one type of therapeutics, the number of drug-like molecules that you may want to look at is 10 in the 60s. There is no way you can go through these molecules in any experimental fashion and try to check them on. So how is drug discovery done today without AI? So it's primarily driven by human expertise and human knowledge. So for instance, if you are a medicinal chemist, you know that certain parts of the molecule, here you can see a gray region, it's maybe some functional group that has certain therapeutic activity. So you would say, I want to assemble pieces together to get molecule which exhibits certain profile. Or if you are a biologist, you would say, I want to understand the biological mechanism underpinning the disease and to understand, which targets I should be looking at and design my molecule which will bind to this target. And because, again, it's all driven by our knowledge, which is far from complete, what we are de facto doing is utilizing a lot of experimentation. None of it is like kind of, written in stone. So you just have some guesses and intuition, and then you run molecules, do a lot of screening, to identify promising compounds. Now, the point is that even though there was a lot of advancement in the recent years in terms of, experimental capacity, and now all these experiments are done by robots, you still can only do, in millions of compounds. There is no way you're really going to explore this humongous universe of possibilities. So as a result, we're pretty much stuck to a tiny, tiny portion universe that we, with our human knowledge, can process. So I want to start by saying that, of course, this is a prime space to utilize AI. And the hope is that you can, with a machine learning method, which would not do experiments, at least at the beginning, which would go through this humongous universe in silico, meaning just running an algorithm, just getting predictions, and dramatically decrease the space of possibilities, and then experimentally test only a small subset of these molecules. And, and as we all know, there is a lot of hype about AI. But I want to say that we actually did some interesting discovery last year, almost a year ago. It's a molecule which has its own webpage. It's called halicin. And this molecule has very strong properties against a variety of pathogens like A. bowmani and C. difficile, which are identified as crucial pathogens by World Health Organization. And here below you can see the link to the Cell paper, which describes in detail the discovery process and also tells you more about this molecule. So, and it's interesting that the area where AI made, one of the, first contributions to discovery is the area of antibiotics. What we know today that there is, any antibiotics that you put on the market, it guarantees resistance is coming. And what the graph shows to you is that we are actually losing the race because the rate of resistance is much higher than the rate with which we put new drugs. And there are very terrible projections about what will happen if our antibiotics are not going to be effective. Even simplest of surgeries today require antibiotics. So even though, it's not the perfect drug, but it enables a lot of types of clinical care. And it's interesting actually to look at, progress of science in this area. You can see there were a lot of major breakthroughs in antibiotic discovery in the beginning of 20th century, like 20s, 30s. And primarily they come kind of mining natural products. We all know the story of penicillin. And then people try to further optimize them with some, synthetic processing. But what happened then, you can see this kind of area of void where very, very few candidates are placed on the market. So we can say that in the last few decades, we barely see any significant changes in the antibiotic markets. And one of the reasons is that pharmaceutical companies, because of the economics involved in developing antibiotics, you take them for the short time, they're cheap. Pharmaceutical company pretty much walked out of this area, and we are unprotected. So if AI can effectively find these drugs, as we did with halicin, and even further improve it, it can really be a game changer in a place where traditional pharmaceutical industry, has not much interest. So let me tell you that when we discovered this particular molecule, it was done really on a budget, on a very small budget, in fact. So what we've done here is we collected a training corpus of 2,500 molecules. And for each one of them, we place them together with E. coli pathogen. It's a type of bacteria. And we've seen which of these molecules kill the bacteria and which ones didn't. So what you see on the left side of the slide, this is kind of a standard plates where the color of each entry shows to you how much reaction happened. Did it inhibit the growth or not? And when the postdoc, Jonathan Stokes, who comes from the lab of Professor Jim Collins at MIT, did these experiments, he not only used, traditional antibiotics, he also used a variety of other like natural products, because we know that some of the natural products have antibacterial properties. He also used, drugs for other diseases and so on. So it was very, very data set. And then for each one of this molecule, when you have a structure, you kind of know what was the antibacterial activity of this molecule. And on my end, because I'm not a biologist by no means, on my end, what I've seen when I got this data was pretty much, these graphs, which is 2D representation of the molecule that maybe many of you still remember from high school. And then the number which encapsulates, which kind of reflects how much inhibitive activity does this molecule has against E. coli. And what you're trying to do, you remember that Alyssa, when she started like teaching you machine learning in few slides, she gave example of cats and dogs. So here it's in some way similar idea. Instead of doing cats and dogs, as I said to you earlier, certain parts of the molecule, it can be some subgraph, it can be several subgraph, correlate with the activity. And by giving to the machine, the graphs and the activity, you force it to learn what are the structural characteristics of the molecule that correlate with inhibitory properties. And what this design enables you to do is totally change the way you are looking at it. You can look at hundreds of millions of molecules that we actually scan very large repository. It's called Zinc, which you can never do empirically, no way, even if you're a big pharma. And then we selected only hundreds of these molecules for empirical testing. And then at the end, we identified very few candidates, and eventually they were tested in animal models. So if we can bring this line of work in all areas of therapeutics, we can make them cheaper and we can make them faster. And, and we really had some kind of beginner's luck, because typically one of the things that people know who work in AI, that, your model kind of fits, it learns the principles that it sees in the training data. And our training data consisted against activity for E. coli. But what happened actually, that this particular molecule also showed very strong properties against other pathogens like C. difficile and A. baumannii. And even more remarkable, it was effective against drug-resistant strains. And one of the reasons is that it had a different mechanism of action. So it was a new mechanism of action that they haven't observed, and model managed, to do the magic, So the key part, capacity that, enables us on the computer science and on the AI and to do this discovery was property prediction. So what you want to do, we apply this idea to antibacterial property, but you can apply it as the question, would this molecule be toxic? Or is it soluble? Or does it have some anti-cancerous activity? It all depends, what kind of training set do you have? So you would have the graph and the number, number would capture what you wanted to capture, and then you train the model and you can make the predictions. And maybe the key change you can ask, did you guys really discover something new? Did other people haven't done that? And of course, there were a lot of people before us in medicinal chemistry who thought about the idea that the model should be able to learn the species instead of using human experts. And typically the way they've done it, they will take the molecule and encode it as a vector, where each vector kind of records different substructures. And then the model kind of learns which combination of these substructures is how it correlates with a property. But here we had a problem. The problem is that it was up to human to decide what are the important substructures. And as we know, human intuition can get you that far. So the modern technology, which are deep learning technology, what they enable you to do is actually eliminate human from the loop when you decide about the representation. So what this model do, they will take this 2D representation, the graph of the molecule, and learn how to create a vector which represents this molecule in such a way that your prediction is optimized. So in other words, and I'm intentionally not getting into details, how is it done? For those of you who do computer science or AI, you can imagine doing it with graph, convolutional networks and others. But what it will do de facto, it will take these 2D graphs and embed them into some high dimensional space. I'm showing you here 2D projection, where the molecules which have similar properties are going to living together. Their vectors would be close, and molecules which are very far apart, which have different properties, will be far apart. Now you can take any new molecule, predict where it is located in the space, and give you the value. And we build this model. It's called ChemProp. It can be very extensive. It has been tested very extensively across, many data sets, both public and private in the pharmaceutical industry. And, it works reasonably well. Now, I want to go back to the question, which is really a fundamental question whenever you're thinking about AI and modeling, because, people always think, my main problem is, which algorithm am I going to use? And of course, there are options. But the key part to understand is actually what is really important is to understand what exactly are you modeling, and then look for an algorithm. So when I started my talk, I showed you this particular formulation, which is kind of actually predominant formulation in the field, is that, I have the 2D graph, I have a number, and we're going to learn the correlation. But for those of you who comes from life science community, it sounds like really huge oversimplification. Where is the biology in the story, correct? Because we actually know this number didn't just show up miraculously. There is a story to it. You have a protein, which is a target. The yellow here on the picture is a molecule. You try to bind them. There is some biological processes going on that people study for a long time. And then you can understand, this whole process. And what I am showing to you, it's true oversimplification. Now, some of you may think, it's not a problem. And I agree with you, it's not a problem, because today, machine translation systems that you are all using on Google, they don't really understand in our degree of understanding English. We're not putting English grammar, they learn how to encode English sentence into a vector, and then from that vector you generate a French sentence or whatever other sentence. But the point is, that there is a lot of deep understanding about physical and chemical properties of this connection, so how we can put them into the model. And at least at minimum, you want to know what is the target that you operate with. Now, the thing that I want to say, if you have a lot of screen data, you actually don't have to do this very sophisticated modeling. If you have a lot of data, you can learn it even if you don't do sophisticated things. Unfortunately, in life, many times we don't have as much data as we need. And one example was actually COVID antivirals. We know that there is no single compound now which has strong antiviral properties. Even remdesivir does better than placebo, but not by much. So many people who work in this field felt that they really need to look at the drug combinations. And we know that for other viral diseases like HIV, combination actually extremely successful. And we were thinking, can we take, combinations of the drug and see if they have antiviral activity? The problem was that we work on this problem with NCATS, which is an institute of NIH. They only had 300 screen combinations. They have individual molecules, but they didn't have combinations. And at that point, we realized that we really have to bring in biology. And there is a lot of understanding what kind of targets, both human, like host targets and viral targets, are involved in this process. It was published very early during the COVID times. So we said, why wouldn't we incorporate this information, at least the name of the target, into the model? So the model that I showed you before is kind of the picture shown here. You're starting with a molecule, you summarize it into vector, and then you do prediction. So now we can augment this model and teach it not only to do the prediction of activity, but also identify which targets are involved, because at least for some, we know what are the relevant targets. So the model can express what is it doing in the language of these targets of the protein involved, and then from there reason about the activity. And now if you do that, you can actually put multiple molecules together and see what do they do together, because you know that you get synergetic effect if molecules have activity by kind of addressing different targets, activating different targets. And we've done this modeling, and you can see that for these drugs, for this particular, for remdesivir, the NCATS group was interested in remdesivir. You can identify other molecules that on their own, they are not sufficiently strong killers of the virus, but when you combine them together, they have synergetic effect, and they can actually fully kill the virus, especially the combination on the right. But there is more to think about it. Again, this is a very simplistic modeling. I just saw this is target, the name of it, and it already helped. But what we want to do, and that's where the group is moving, is really bring heavy biology into the process. For instance, molecules actually, they are conformers that live in 3D space. So it may happen the two graphs are just different by one molecule, but by one atom, but they have very different properties, because in 3D they look very differently, but we don't see it. So we're trying now to model these conformers and to model the protein structure to make better predictions. And the final thing that I want to say is that, this number 10 in the 60s, even if you use a cloud and you have a lot of compute, you still don't have enough capacity to screen 10 in the 60s. So what we are doing now is actually building models that can de novo generate your molecules. You just say, I want these properties, and we're going to generate your molecules with these properties. And it's very much copies what medicinal chemists do. We first teach the model to identify the pieces, kind of the alphabet of each property, and then we combine them together to create these molecules, and it works, pretty well. And here you can see pictorial representation of that. So you kind of do your property prediction, identify what are the pieces that are likely the one that are driving the property, and then you take these pieces, and it's like a Lego, and you assemble your molecule from these Lego pieces, then to generate it to create a correct chemical structure. And this is, the area of very active research, both at MIT and, other places which do machine learning of molecular structures. And I want just to finish my slide by saying that, I'm very realistic. We are really in the very, very early stages of what AI can do in drug discovery. There is a very long road to go. But when we are looking, at all the, I just put some, illnesses for which we don't have very effective drugs, for many of which we don't have effective drugs. And I think AI can really change the game here. Thank you.
Thank you. That was wonderful. And I think that we got a very good view of a fair swath of the landscape between what Casandra and what Regina just said. And I just want to ask one question to Regina, and then we'll start the joint discussion. And Casandra, Regina, you obviously can ask each other questions too, and we'll take some questions from the audience after we talk for just a few minutes. So my question, which actually was also raised by someone in the audience, was those 2D models that you said that we saw in high school, et cetera, are quite different than the 3D models. And actually, I realize this is a question for both of you, but I'll ask Regina first, and then Casandra can talk about it. So I mentioned before that I work in data visualization, and so I know how important and hard it is to connect 2D to 3D representations. And so in your talk, Regina, you mentioned that the geometry turned out to be really important, and then you essentially found these like hidden predictors in the geometry. And so the question in the audience was whether that was from 2D or 3D. I think your answer is that it was from 3D. But my question for both of you is, how do you like encode that into machine learning algorithms? Because like the images that are published often don't have the 3D structure. You have to describe the 3D structure in some way. Like, is there a way to translate 3D representations into the graphs that you needed to use, Regina, and then into something that Casandra would use? Like, can you just tell us more about that?
So we are currently, this is actually a very interesting piece, that despite the fact that, everybody are thinking about the importance of using 3D, I would say that in machine learning models, we haven't seen 3D, really working better, significantly better in all data sets. In comparatively 2D. And what we need to understand that one 2D graph results in multiple conformers, in multiple 3D conformers, and depending on the protein, you would select the conformer which does a binding. So there is also a probabilistic aspect. So look at translating 2D to one 3D, there is a whole distribution of them. Right. And there are lots of ways to do the encoding. People are exploring different mechanisms to encode. There are software that can actually deterministically, talk kind of reason about the angles, and you can fully encode. But the problem is that in most cases, we don't know which conformer was the one which resulted in the binding. You need to model it as a hidden variable, and and sometimes it works, but many times it doesn't. So there is very hard computational questions.
And then thank you. And then for Casandra, is there enough of a language about that in the literature, and enough of a standardization in the way that that's represented that if that 3D geometry actually did turn out to be useful, that the BenchSci approach could actually capture that? Or is that something where you have to sort of have a whole add-on effort to try to think about how to record a sort of new description like that?
Yeah, that's a great question. I have to agree with Regina on this. We are still at early stages. It's not just about the interaction. If you think about the central dogma of science, you have your DNA, which gets transcribed to RNA and then into protein. Even then, there are challenges associated with that. A lot of target identification in terms of understanding the baseline of disease is dependent on genomics analysis. But if you think about all the steps that it takes to get to a protein, how that disease actually occurs, all the different, what they call omics between that, how the DNA is modified, how the mRNA is modified, how it actually works in a more complex environment when you have, acid in the stomach. All of those things are part of the biology that it is very challenging, even for a human scientist to understand, let alone a machine learning algorithm. So I think it's about leveraging machine learning to date, leveraging what it can do to this point, and then supplementing that with human knowledge. And then as the technology progresses and we have better information, better data sets, better technologies, we can ultimately one day get to that point, I think.
Yeah, and we had a question from my co-director in science from Immaculata de Vivo, who's an expert in these matters, and she asked me to ask you, Casandra, with so much variability in lab experiments, whether there were certain experiments that were better to be harmonized with others. And I guess that also translates, Regina, into a question for you of like, are there particular sources of data that turn out to be the most useful and you want more of those? And then just to make this question a little bit more complicated, many people in the audience have been asking about whether or not scientists themselves would ever sort of tag their data or tag their articles or tag their figures in ways that it would make it useful, more useful to you to use the information that gets published and distributed. So those questions are sort of related. It's sort of like, how do you find the most useful data? And would it be practical? I have my own opinions on this, but would it be practical to ask people to apply some sort of tagging or rules or standardized language?
Yeah, so maybe I can start that
Casandra,
one off.
You can go first. Yeah.
So definitely there are indicators within experimental data that human scientists are looking for to determine the quality of an experiment. How good is the signal? How good is the stain? What are the steps that went into actually processing the sample? So all of those things are a part of the evaluation of the experimental output. And then there are some things that we could use as indicators. I think the biggest challenge right now is actually finding those experiments that are buried deep within those data sources. So there are, many of the things that we hear is, if I had only known that paper existed, I could have made a more informed decision about my experiment going forward. And sometimes those papers actually are not within your field of interest. So being able to surface those experiments and have the human scientist evaluate that first is usually the first step. And then there are additional things that you can do even outside of machine learning to tag that information to understand quality, was that paper retracted? Is there a reason why that paper got retracted? Is that particular experiment responsible for that? How much rigor went into designing that experiment that you could ultimately tag on those images and make it a part of, what we would do on our interface to evaluate that quality and have scientists determine that. So I think the idea here is to tag as much as you possibly can that's scientifically true, and then provide flexibility through interfaces to allow the scientists to determine what defines the quality so they can be returned results that are actually the ones that are most important.
Yeah, that's great. There were a number of questions you just answered without knowing it in the thread here. The number of questions about quality and about limiting the training set and expanding the training set, and I think you just answered a whole bunch of those. So let me change the topic just a little bit. The audience is also quite curious, as am I, into the public-private partnerships that are kind of implied in your work and present, obviously, in your work. And so, Regina, you're at an institution of higher learning, MIT, and Casandra, you're at a commercial company. But obviously, you're both relying on work and research and publications that have been made in both of those arenas. Funding comes both from the government and from private sources. And some data are open and some software is open and some is not. So what do you see? I'll ask Regina if you would like to start. What do you see as the sort of ideal world and combination of openness and public-private cooperation in an ideal world? Okay, you can also give me a practical answer, but I'm asking you in an ideal world as well.
So when you asked me a question, I realized I didn't put my last slide. So actually the tools that we developed the whole bunch of tools. They are distributed because this work is supported by MLPDS. This is a machine learning for pharmaceutical discovery and synthesis consortium, which has, I think, 16 major pharmaceutical companies, which are paying for the development of these tools and which are using these tools in, in their daily life, which enables us to understand how they work for real in drug discovery. So in our case, the way we are doing all these tools, they are made public. They're not only used by these pharmaceutical companies, but they're used, with broader in the industry and in the research community. I think it's really crucial to make this software open to create benchmarks. Now, the part where I think we have some issue with, privacy, private-public connection relates to the data. So majority of the data that we are using is like massive data sets that are, that are screened. We don't use data extracted from the literature. Literature doesn't have this data. and somebody needs to go do the screening and put this data out. And what you can see, how what is available actually impacts the research. On one hand, you can see that most of what is done in AI today is done on small molecules, because small molecules have a lot of data that was screened and put in public. On the other hand, biologics, for them, there is no public data, and as a result, there are very few AI techniques developed for processing this type of molecules. So you can see that on one hand, of course, every pharmaceutical company wants to keep the data to themselves, which is great. But on the other hand, it really stops the development of the certain branches of discovery, and at the end, I would say it doesn't really give much benefit to the companies who hold this data but don't have the right tools to process it. So I am still kind of looking what is the right way to make this data available. Yeah.
Thanks. Yeah. And Casandra, that would be great if you could answer the same question. And while you were talking, there were some more questions about public repositories and how those work. And so if you can throw that in, that would be great.
Sure. So I am a huge proponent for open access. I completely agree with what Regina just mentioned. Even just looking at data sources like publications, some of the access that we have is closed access. We have partnerships with those publishers. We actually get access to the full content so that we can apply our algorithms, and then we point users back to the paper. One of the challenges, especially I faced in academia, is that you don't know if that paper actually has the information that you need, because all you have access to is the title and the abstract. So one of the ways that we help break down that barrier is to analyze that ourselves. Scientists can type in exactly the parameters of their experiment they're interested in, and they'll know that paper has the content or the experiment that they're looking for. So at this point, if they have subscription, they can access it, or if they want to purchase the article, now they have more confidence that that data source has what they need. But I do think there is a movement towards having that data open access, even repositories like bioRxiv, where data is being input as early as possible so scientists can benefit from that knowledge. I mean, the entire premise behind BenchSci is to leverage the world of scientific knowledge. And if that's buried, right, how do we help move science forward? Right. And
I think the world is much more familiar with bioRxiv this year thanks to COVID than they would have been
yes.
Before. But yes, there's an unintended good consequence of COVID. But speaking about consequences and fair and unfair consequences and good and bad, just to bring up two examples in the questions again, there was somebody who mentioned what the impact of AI on orphan diseases and rare conditions that might not otherwise be researched would be. And I'm guessing that your answers are going to be that it's much more cost effective, and so that's a kind of a selling point for AI. And then of course many people are interested in the accidental or purposeful, but I think mostly accidental introduction of bias into any of these procedures, just because of accidents of what the training data allow, or even what the humans are interested in. And so this isn't necessarily racial bias or gender bias in the kind of work that you're talking about, but it could be, disease bias or molecule bias or lab bias, or, and then accidentally translate into these other things. And so if you wanted to sort of talk about where the really great potential for AI is in the future and where the really dangerous potential that we have to think about is, can I ask each of you to answer that question? And this time, let's start with Casandra.
Sure. So in terms of bias, I think it's very difficult to eliminate it completely. I think there are definitely efforts whenever you're developing to make sure you're minimizing that as much as possible. And I'd love to hear what Regina's thoughts on this are as well. When you're looking at training sets and you're trying to develop those for the purpose of training your algorithm, sometimes you're limited by what you have access to. Sometimes it's very important the training set that you choose. There are certain repositories for specific cancers. There are certain publications that you might want to look at. One of the things that we want to ensure is that when we're looking for proteins, applications, diseases, we're not limiting that in any way by the data source. We're looking for everything, everywhere. It's very objective. Was this protein studied in this image? And what was present around it and what context was it studied, and when we surface that. If you look at the interpretation of the scientist as part of your training, then I think that creates bias because you're looking at the result and the interpretation of the scientist in that sense. But when you're looking objectively at how the data was reported in the image, then you're just looking at what was there. And in doing that, you actually open up several opportunities. You may not realize that a scientist studied a particular protein for a disease, and that protein is actually very important to another scientist and had no idea it was associated to that disease. So by looking at things very objectively and extracting that and just surfacing that, I think that's the tremendous opportunity, because one of the questions that we often get is, are you removing the serendipity from science by pulling that data and characterizing it? But in fact, you're bringing visibility to that data, letting scientists know where those proteins have been studied in the past in a way that they may not have known because they limited themselves by only looking at certain publications and certain data sets. So I think in terms of the possibilities, by doing this objectively and making sure you're doing your best to eliminate bias as much as possible, that is really how you address it going forward.
That's fantastic. Thank you. Thank you. And it's a sort of subtle point too, because I think not a lot of people realize that AI can be applied to the literature in that way to extract data and information. And what you're basically saying is you're using all of the literature that might not otherwise be exposed. And so that's likely to reduce bias at some level. Yeah, thanks. Regina, your thoughts.
So I think that the bias here is actually the human knowledge itself, because the data that is available for you to train is the data that some human elsewhere decided that it needs to be screened. And it doesn't matter, they published it, they put it in their repository, somebody signed that these molecules are going to be bought and screened. And they didn't do it randomly because it's very expensive. They use their prior knowledge. Now, what you want to do when you are working in this field is not to do more of the same, because they can buy more of the same molecule. You really want to jump to new parts of chemical space where we didn't explore, where we don't know what will happen, and maybe there are some miraculous molecules. So to me, the solution is, there's a solution which has like two parts. The first one, we need to develop algorithms which can generalize correctly and can jump in different distributions. So this is like a big area in machine learning. People look at invariance-based representation, which can kind of generalize along the right axis, and there will be more of this work. It's the same algorithms that help you to do well, recognizing images of different population. You can think about, developing this methodology also in the molecular space. We do that. And the second one is to change the way of thinking about, how the data is collected. Today, which is the first generation of the models, you first, somebody again bought this molecule, screened them, and then they give it to me and say, build me a model. But in reality, it's very wasteful, because maybe we don't need this data, we need some other data. So you can imagine much more fluent process where the model can tell you what it actually needs, and jointly with the model and the human, you identify how to sample training data to get to the best outcome. And if I might just say, briefly look at the question, there was an excellent question there. I don't know that the others were not excellent, but one question about how AI can change, the kind of orphan diseases and tropical diseases. I think it really has a huge potential, because this antibiotic that we created, which is a very strong molecule, it cost, less than, a few $100,000. It's nothing in this world of drug design. And we are currently working on diseases like Chagas, which is a big tropical disease, which affects, millions of people without good cure, and many others. You can just apply this methodology to scan huge space of possibilities, identify a small subset, and then test them experimentally, which is expensive. You can predict ahead of time what kind of bad consequence your drug may have, so that you don't need to run the whole clinical trial to discover it. So there is a lot of capacity to be better in prediction, the same way as introducing e-commerce really cut a lot in terms of supply chains and other things. I believe AI should do the same for all these diseases that are currently hard to work within the traditional pharma.
That's fantastic. Yeah, I mean, this is really good news. And I just want to mention a couple of events that are coming up at Radcliffe, and then I want to thank you both and ask you one concluding question. And so I just want to say for everyone who's listening, and you can find this on the Radcliffe website, on May 11th at 4 p.m., There'll be a webinar called Vaccine Equity and Efficacy in the US and the World, which is clearly related to today's discussion at some level. And then even more related, this is kind of the first event in an AI series that Radcliffe is creating for the coming year. And there will be a symposium on October 22nd that may be all day at Radcliffe, depending on how the COVID thing goes, or it may be online. We'll see what happens. And then again, that's on October 22nd, and it'll be on topics related to artificial intelligence. And so just to close out here, I just want to ask you what you think should be done about education in schools, in colleges, and in the public around AI. Because as you said, Regina, there's a lot of hype around AI, and I think a lot of people don't completely, think it has to do with killer robots, and that's sort of the end of it. And I am being somewhat facetious. I think most of the people joining us today do not think that. But in all seriousness, do you think that computer science, do you think that AI, do you think that this kind of reasoning, what do you think should be taught in school so that people can make, informed analyses and decisions as members of the public? So Regina, why don't I start with you, and then we'll go to Casandra. Just a quick answer, because we're almost at the end of our time, but thanks.
The quick answer, I actually think that today, if you are interested in AI, there are lots of places where you can take AI. I think in order to make real progress in this area, it's not only having AI education, but it's truly about being bilingual. And when I started working in this area, I'm still, learning a lot. So, to help with it, we created now a course at MIT, which is called Modeling with Machine Learning, which actually is not for computer scientists, which have enough of their machine learning courses, but really for all the students in science and engineering at MIT, which can learn the fundamentals of modeling with AI, and then they have sections, let's say, in chemical engineering or in other areas where they can apply them directly to their area.
So by bilingual, you mean AI plus some domain that where it may
AI plus some domain. I think it's the same way as we all are using computers today without being computer scientists. That's, I think, where we need to go with AI.
Yes, we are. Thank you, Zoom. Okay, thanks, Regina. And Casandra, what are your closing thoughts on that?
Yeah, I truly believe that AI is the future, not just in healthcare, but in every other field, and education is a huge component of that. I think there's apprehension around the application of AI, leaving it unchecked. But I think if you learn more about it, you can certainly understand the benefits and how powerful it can be in every industry.
Well, thank you. And I thought this was just a wonderful conversation. And I thank the world of the Zoom gods for having it continue to work. And of course, I thank the Radcliffe Institute and all of our sponsors. And I hope that you'll join us for the 2 future events I mentioned earlier, the COVID one on May 11th, and the COVID vaccines rather, and then the symposium next October, and that'll be on the website. So thank you, everyone. And you can find the video for this online soon. Thanks.